Great. As people make their way back from the coffee break, we'll get started with our second session. So the topic of the second session is cancer and genomics, and as many of you have probably heard, genomics technologies have really expanded and evolved tremendously in the past decade and recent years, and one of their applications has been in understanding cancer, and so we thought to combine these two topics in one session. And today we're going to hear about amazing talks from both campuses, actually, both Morningside and Medical Campus, on drivers of pancreatic cancer, high-throughput, large-scale screens, studying responses to therapies, how diet and metabolism changes the fates of cells, and post-transcriptional regulation of gene expression. And our first speaker, we can start with Dr. Christine Chio. She's an assistant professor of genetics and development and in the Columbia University Irving Medical Center, and her research focuses on the role of reactive oxygen species and redox metabolism in cancer, and one of her notable contributions is the elucidation of how reversible cysteine oxidation influences cancer cell survival. She's supported by many awards, including R01 Awards and the DoD Cancer Development Award and the Pershing Square Prize, among many awards to name. There's a long list. So without further ado, let's welcome Dr. Chio.
All right, good afternoon, everyone, and thank you for the kind introduction. Thanks for having me today. So my lab studies metabolism in cancer, and a specific question that we ask in the lab is, what do reactive oxygen species do in cancer? This is a question that has been around for a long time. There are many different sources of reactive oxygen species, extracellularly from UV ionizing radiation, intracellularly due to changes in metabolic programs within the cell. And over the years, the field has largely asked the question of reactive oxygen species through the lens of oxidative stress. And what this means is, what happens to cancer cells when there's too much reactive oxygen species? What are the cellular programs that underlie the stress response as well as cell death? So in addition to the two ends of the spectrum, where you have too much or too little reactive oxygen species, our lab is interested in asking what happens in the hormetic region. What is the role of reactive oxygen species in addition to its indiscriminate functions of killing and damaging cancer cells? So what do I mean by this hormetic region? So if you think about it, the major targets of high levels of reactive oxygen species, it's DNA and lipids, and we will hear from Brent later on the role of lipid oxidation and ferroptosis. However, the major when reactive oxygen species are in the hormetic region, the major biological target, it's not DNA or macromolecules like lipids, but instead the major targets are amino acid residues. And some of the amino acid residues are shown here. All of them can undergo oxidative modifications, meaning when there are changes in levels of reactive oxygen species, you either get a reduced or an oxidized version of each amino acid residue. Much of our previous work has focused on characterizing the cyclic reduction and oxidation of the amino acid cysteine. And here we have shown in the context of cancer that these cyclic oxidation events control protein synthesis programs in order to promote tumorigenesis. But for today's talk, I want to focus our attention on the other sulfur-containing amino acid residue, methionine. Now, to most of you, methionine is a hydrophobic amino acid residue, and also it's the first residue of a polypeptide chain. But to us, methionine does a lot more. Just like cysteine, methionine has a sulfur atom. What this means is this allows methionine to cycle between its reduced and oxidized state. But what is special about methionine is that the sulfur atom forms a chiral center, which means upon oxidation, you either get a left-handed version or a right-handed version of the sulfoxide of this residue. And more importantly, the reduction, meaning bringing the oxidized states back into the reduced state, it's mediated by specific classes of enzymes called methionine sulfoxide reductase. There's one enzyme that is specific for the S-diastereoisomer, and there's an enzyme that is specific for the R-diastereoisomer. So my argument is nature will not design specific enzymes to reverse the left versus right-handed version of an amino acid residue unless there is an important function. So what's the evidence for that? We studied this in the context of pancreatic cancer, and we have seen using patient-derived specimens of normal, pre-malignant tumor and liver metastasis from patients that the expression of this reductase that I mentioned in my previous slide, it's decrease very significantly as the disease progresses. This can be observed in the context of tissues, and we can also see this in organoid models of pancreatic cancer. And here I'm showing you again a stage-dependent decrease in the expression of MSRA, this reductase. And notably, the other family member, MSRB, which regulates the reversal of the other diastereoisomer, does not exhibit a similar decrease in expression pattern. So this tells us that site-specific changes in oxidation patterns of methionine residues contributes to the metastasis of pancreatic cancer. But is there functional evidence to support this idea? We went on then to take our organoid models and perform CRISPR-Cas9 to knock out this reductase, MSRA, in pancreatic cancer cells. We then injected these cancer cells into the pancreas, and we asked what happens to primary tumor growth. And here we saw that there is no significant difference in primary tumor burden. However, in mice that received cancer cells that lack this reductase, they exhibit a significant increase in the incidence of metastasis. And if we were to focus on liver metastasis, where we inject the same cells, but now through the spleen to ask how many of these cells can migrate through the portal vein and seed the liver, we now see an even more striking difference. Loss of this reductase significantly increases both the size and the number of lesions that we find in the liver. So our genetic data tells us that loss of this reductase, loss of the ability to reduce methionine residues, promotes pancreatic cancer metastasis. But our main interest is in understanding what are the downstream mechanisms. How does the change in the expression of this reductase have such a profound effect? And so we asked whether we can address this question globally using proteomics. In 2017, Chris Chang at UC Berkeley reported that oxaziridine, an N-transfer compound, is able to selectively derivatize reactive methionine. And so we asked if we can leverage this chemical reaction towards the development of a probe that can help us separate fractions of the proteome that contains reduced methionine versus parts of the proteome that contains oxidized methionine. And so we teamed up with Chris's lab to synthesize a panel of oxaziridine analogs, and we confirmed that indeed oxaziridine, these analogs are selective for methionine residues, but not for other amino acid residues. And now we ask if we can further add an alkyne handle. This allows us to perform click chemistry so that we can add a biotin through copper click reaction. And so all reactive methionines, as you can see, will be derivatized and will contain a biotin group. This means we can then perform streptavidin enrichment followed by proteomic analysis. And using this approach, we were able to characterize the proteome of pancreatic cancer cells at different stages of disease progression. And through these efforts, we found that In a metastatic setting, there are proteins that undergo selective reversible oxidation. And one such protein that we characterized was an enzyme called PKM2. And what we found was reversible oxidation of this methionine residue switches PKM2 from a dimeric to a tetrameric state. And this mechanism controls whether cancer cells exist in its proliferative state or switches over to a bioenergetic migratory state. And we think this mechanism underlies the process of metastasis. So how do we go about translating this information for therapeutic development? What I've told you so far is this enzyme, methionine sulfoxide reductase, decreases in expression during the course of pancreatic cancer metastasis. And so if we are able to bring back the expression of this protein, in theory, we should be able to curb metastasis. And so we asked whether we can first do this genetically. And so the approach is to use a DOX-inducible system to restore expression of MSRA. We then introduce these cells, again, through the spleen and assay for liver metastasis. And interestingly, what we found was when we restore expression of MSRA, this significantly decreases the number of liver metastatic nodules. But more significantly, while every single mouse in the control group developed extrahepatic metastasis, not a single mouse in the DOX-treated group developed extrahepatic metastasis. So this genetic experiment tells us that if there is a chemical way for us to bring back expression of MSRA, we are in a position to mitigate metastatic dissemination. So how do we do this? Some of you in the audience may be familiar with the concept of PROTAC. The idea of PROTAC is to develop a heterobifunctional molecule that allows you to bring together your protein of interest with an E3 ligase. This allows for targeted degradation of your protein of interest. We ask if we can do the reverse. And so to this end, we're developing a system called DUPTAC, which involves development of heterobifunctional molecules that now brings together our protein of interest with a D-ubiquitinase. And so here are the compounds. This approach allows us to synthesize compounds that are highly modular. There's a part that is recruiting the D-ubiquitinase. There's a linker which allows us to switch out the bait. And in here, the bait is methionine sulfoxide, which is expected to recruit MSRA, our protein of interest. And so this is an ongoing project that we are pursuing in collaboration with Santi at the BME department. And the idea is to also, in addition to developing these DUPTACs, partner with Santi to develop nanoparticle-based hydrogels for targeted delivery to the tumor. And so since our initial efforts to characterize methionine redox proteome changes within the cancer cells, we have expanded our efforts to characterize changes in organs and tissues outside of the tumor. We know that cancer is a systemic disease. In addition to changes detected within the tumor proper, every single organ within the body changes when a person or a mouse develops cancer. And so we have since characterized all tissues in our mouse models and also patient-derived specimens comparing non-tumor to tumor-bearing subjects to characterize what are the changes in the reactive methionine proteome as well as the cysteine reactive proteome. I wouldn't have time to go over the details, so if anyone's interested in talking about this, please reach out to me later on. But I can give you a little teaser that in both mouse and human with cancer, they exhibit significant changes in their adipose tissue methionine reactive proteome. And we think that this underlies the process of cachexia, which is a debilitating metabolic syndrome that leads to death of a lot of cancer patients. And so if you are a trainee interested in the intersection of redox biology and cancer, and if you're interested in applying chemical engineering tools to develop both diagnostic and therapeutic molecules for pancreatic cancer, please reach out to me. And last but not least, the most important slide, this is my team. Nothing I told you just now would have been possible without these wonderful people. The work today that I presented was spearheaded by James and also Eugene. We have a lot of support from collaborators within Columbia and also outside of Columbia, and of course my funding sources. Thank you so much for listening.
So last but not least, we have Professor Brent Stockwell. He's a professor of chemistry and pathology and cell biology, William Keenan Professor, and chair of the Department of Biological Sciences. Dr. Stockwell is really a pioneer in the field of chemical biology, and he discovered and coined the term ferroptosis, which is a form of cell death. He's a member of the National Academy of Medicine and received numerous awards, including the Beckman Young Investigator Award and HHMI Early Career Scientist Award, NCI R35 Outstanding Investigator Award, among many others. And he has been in the top 1% of highly cited researchers in the last 4 years and was named one of the 50 most influential life science individuals in New York. Brent?
Great. All right. It is a true pleasure to be here. I love the engineering school. The engineering school is such an important part of Columbia. I found the kind of expertise and collaborations we can have. So I just appreciate being here. So let me tell you about some of the recent things that we're thinking and some of our new directions. So we're thinking about metabolism, as we've heard about metabolism today, and about metabolism as being in many ways at the center of cell function. And we always think about the central dogma. We hear about RNA and DNA and protein function. But I think in some sense, metabolism can be a layer on top of all of that, and really can control gene expression, protein function, and ultimately cell fate. And so that's what I'm trying to show you here, that metabolism can affect all of these different molecular processes, and metabolism in turn is controlled, in some cases at least, by diet. And so that's an interesting direction we've been looking at. Can we use diet to control all of these kinds of processes? And in particular, we've looked at this in the context of ferroptosis, a kind of cell death that we identified about a dozen years ago. And it lies at the intersection of metabolism, redox biology, and iron biology, and kind of integrates those fields in terms of controlling this process, and it's involved potentially in other physiological and pathological processes. So the question here is, how can we leverage metabolism, particularly through diet, to control cancer cell properties? In order to really answer that, I would argue that we need a blueprint that connects metabolism to cell fate. And so for the first time, I think we have that, a really nice blueprint in the context of ferroptosis, this kind of metabolism-linked cell death. We're understanding more and more through the work of many labs around the world how the molecular regulation of metabolism connects to ferroptosis. And so we can try to use that. So this is just showing you that we identified this about in 2012 as a distinct form of cell death, and a lot of this was led by my former postdoc, Scott Dixon, who now has his own lab at Stanford. We identify it as this iron-dependent oxidative form of cell death. There's just to say there's a lot of papers on ferroptosis now. If you haven't heard of it, hopefully you've heard of it now, but maybe not. Anyway, there's 163 NIH-funded projects on ferroptosis for over $61 million. And so let me turn to this question of diet and how it might be relevant now that we have this blueprint to understand and control ferroptosis. So I'll give you one simplified view of ferroptosis and metabolism, which is that in order for cells to die through ferroptosis, they need ultimately to get these oxidized lipids that drive the cell death. These are phospholipids that have a polyunsaturated fatty acyl tail, so we call those PUFA phospholipids. And when they get oxidized to this peroxide, that becomes a lethal event for reasons that we still don't entirely understand. So in order to get these oxidized phospholipid PUFA molecules, we need two things. We need to have the PUFA incorporated, obviously, into the phospholipid. So the level of PUFAs will be important. And then we need some iron-dependent enzymes and labile iron that does the oxidation. So the level of iron will be important. And then there's a complex set of factors that can repair these oxidized lipids, particularly this enzyme GPX4, glutathione peroxidase 4. It uses the peptide glutathione as a co-substrate. And glutathione is in turn built from cysteine, which we heard about from Christine, and methionine also. So the question is, would the level of these factors in diet potentially influence the sensitivity of different tissues and tumors to ferroptosis? So we started looking at this with Peter Knoll, a neuropathologist at the medical center, in a glioma model. And I'll summarize a lot of experiments with two figures that we did with Peter. One is we just took his mouse glioma model and put them on a diet that was depleted in cysteine and methionine, which we know are needed to prevent ferroptosis. So we thought this cysteine methionine depleted diet might enhance the sensitivity of these tumors to ferroptosis. And actually what we see is just putting the mice on the diet alone with nothing else actually does improve their survival. So suggesting that there is some benefit. But if we come in now and treat the mice with a compound that inhibits that enzyme GPX4 that I mentioned, this compound's called RSL3. It's delivered through a convection-enhanced delivery method that Peter and his collaborator Jeff Bruce developed. Then we see that there's now, in the context of the diet, the cysteine methionine depleted diet, a really pretty significant improvement in survival, so that the diet plus the drug does have a benefit in this model. So then we turn to that other part of that diagram that I just showed you, the level of iron and the level of polyunsaturated fatty acids in diet. And in this case, we turned to a lymphoma model because we found that certain lymphomas are very sensitive intrinsically to ferroptosis. So the question was, could we change the level of PUFAs or iron in the diet and affect the sensitivity of these tumors to ferroptosis? And we did see that. So this is a xenograft model. So we took a diffuse large B-cell lymphoma cell line, and xenografted it into immunocompromised mice in this case. And then if we put them on a normal diet, normal mouse chow, then the tumors grow up as they are wont to do. But if we give these mice either a high iron diet or a diet high in polyunsaturated fatty acids, it's a mix of two PUFAs, then we see a pretty significant inhibition of the ability of these tumors to grow. Now, of course, that could be due to many different effects, one of which would be ferroptosis. There could be other effects as well. So to test how much of this is due to ferroptosis, we did the same experiment, but then we also treated the mice with a small molecule inhibitor of ferroptosis that prevents that lipid oxidation that I talked about. And that's shown here. So now if we treat with the inhibitor of ferroptosis in pink, we see we restore the normal growth to the tumors even in the presence of the high iron diet or the presence of the high PUFA diet. So it does seem to be through this mechanism of lipid oxidation and ferroptosis. And then another key question is, can we predict which tumors in the end will be sensitive to this kind of diet or to ferroptosis? And the work of Bayu Chu in the lab has answered this in some sense. The best predictor we found so far is actually not a mutation or protein change, but actually the presence of a lipid, which makes sense. And this is actually a rare lipid that we identified as being now a key driver of ferroptosis. It's a phospholipid that has two polyunsaturated fatty acyl tails on it instead of the normal one. Most phospholipids have 0 or 1 PUFA tails. So we found this unusual class that has 2 PUFA tails, these di-PUFA phospholipids, and they seem to be great predictors of the sensitivity. These are different cancer cell lines, and their level of sensitivity to this ferroptosis inducer. And so the more of these di-PUFA lipids they have, the more sensitive they are, basically. And then the last question is, can we do this in people? And so maybe you might worry that the doses we were giving the mice, the PUFAs, for example, would be just not achievable in humans. And actually, it's quite feasible. So the mouse PUFA dose is about 250 milligrams per kilogram we're giving every other day. So if this would be like a high PUFA diet where, various normal foods, basically, normal healthy foods, but picking the right ones, you could get up to over a gram a day of PUFAs. So basically, we think this can be done with a customized diet in people, and we'd like to move forward and test that in some clinical trials. So just to summarize, I think diet offers an additional lever for controlling tumor ferroptosis, growth, and metabolism, and we'd like to try to test this now in people. And I'll just thank again the members of the lab. So Iman Ahmed was doing all the DLBCL lymphoma diet studies. She recently defended her PhD. Josh Yu is an undergraduate who is helping her with GWEN, and Jenny also helping. And then Bayu identified those di-PUFA phospholipids as being key drivers of ferroptosis and key predictors of sensitivity. Thanks again for the invitation.
Thank you so much, Professor Stockwell. So now it's time for the Q&A. This is why Alexis prints out schedules for us. If I can ask all the speakers to come to the front.
This is for Dr. McFarlane-Figueroa. For your technology, I imagine that would be really valuable to a lot of pharmaceutical and oncogenic companies, like industry. Have you done any kind of collaboration with them in regards to their potential drugs, or are you interested in having your technology be applied to their drug discovery pipeline, that kind of thing?
So we're definitely interested in the overall, you could see this how it would fit into drug discovery more broadly. We haven't gotten to that point yet. It's to be done, I guess. That's it.
Other questions?
Hi, this is kind of directed a bit more, Christine, at your research. I was just curious as to why you were trying to do some DNA editing in order to promote, I'm going to mess it up, but the MFSA?
MSRA.
MSRA, thank you. Instead of just trying to directly inject it either into the bloodstream or to the local site?
You're asking why, sorry, can you repeat your question? I want to make sure I understand it.
Yes, so you were trying to promote MSRA through, it seems, some form of genetic editing, and I was just curious as to why you didn't just try, or why you decided not to just directly inject it into the target areas or into the bloodstream.
Are you referring to our experimental strategies to knock out MSRA? That was genetic, but the current approaches to restore MSRA is small molecule base. That's not genetic.
Okay, I probably misunderstood it then. But that was more of what I meant. Trying to restore it, why not just have treatments of being injected instead of... I was just curious because it seemed like a lot of hoops to jump through, but I was just curious if there was a reason why not.
So as in why we took a chemical approach as opposed to gene therapy?
Yes.
Okay. We can certainly take a gene therapy approach. There is a part of the story that I didn't have time to talk about. The downregulation of MSRA does not happen at the genetic level. So the gene is there, the mRNA is there, there is an abundance of the transcripts. The problem happens at the post-translational level. So it turns out that there's upregulation of an E3 ligase that directly degrades MSRA, and therefore taking a chemical approach to restore stability of MSRA seems to be the most appropriate in this setting.
I have a virtual question for Dr. Stockwell. In the glioma patients, how much cysteine is given? And second, how are you monitoring whether ferroptosis is actually occurring in these patients? It's right there. They work, they can hear you.
Yeah, so that was in the mice we were looking at in the glioma mouse model. And so we did look to see if the cysteine, it was a cysteine methionine depleted diet. So in other words, we weren't giving them extra, we were taking away cysteine and methionine in the diet. It wasn't 100% depleted, but it was based on what was achievable in the chow. And we did check to see if there was a depletion in cysteine in the tumors, in the mice, and also a depletion in glutathione, which cysteine is used to make. And we did see a depletion in both of those. Whether in that model, we didn't check to see if the effect could be reversed with the administration of the ferroptosis inhibitor, because that's a longer-term model. So that would be a good thing to check in the future.
Great. Other questions? Maybe I'll ask one for Dr. Zakara. You mentioned M6A, its role in AML. I'm wondering if there's a broader link to, other cancer types that have also blocked differentiation, or you also showed that it's MYC-related. Is it dependent on mutations and amplifications in MYC in other cancer types?
Yeah, so it's been shown, in 10 years it's been shown to have an impact on a lot of different cancers. And it seems that there is a pattern. There is always an increase in M6A, probably due to an increase of the enzyme that is responsible of adding that modification. And in the case of neuroblastoma, there is a connection with MYC amplification. We are trying to understand why. We think that MYC can control some of the factors that are implicated in M6A deposition. So it's controlling probably metal 3 or the YTHDF proteins. But we don't know completely everything. We know that if we deplete this protein in neuroblastoma that is not MYC amplified, it's actually making the neuroblastoma even worse. So there is definitely a connection there.
Very interesting. Yes.
I was wondering, how much is the half-life?
Yeah, so the half-life of the M6A RNA is 2 hours, 2 to 4 hours. So it's very, for some mRNA it can be even shorter compared to the long-lived RNA. So the idea here is that if you are in a cancer situation, then what happens is that you have the accumulation of this transcript that they're not now around for just 2 hours, but they're around for much longer, and so they can make protein that are, that are toxic, or they're supposed to be not as abundant, and so they contribute to the disease. That is the idea.
And did you see the concentration, the density?
So we know that the more M6A you have, the shorter is your half-life. And we can measure it using different approaches. Yeah, so we've seen it with different type of methods. Yes. You had a specific question.
Yes, regarding the depletion and the phenotype. So.
Yeah, timing. Yeah, as I mentioned, it can be very, very short, and the more you have, the shorter your half-life will be. We don't know exactly at this stage which sites are more important, right? So as I mentioned, M6A can be in the coding sequence, can be in the 3' UTR. We don't know exactly at this stage which one are more contributing to the half-life. And we hope that these quantitative methods that we are now using can help us understand much better those mechanisms. One example is, for instance, that we think that some of these M6A in the coding sequence are somehow recognized by the ribosome as stalling the ribosome, so and send for degradation instead of making the protein. That could be one mechanism, but we're still in there. There is a lot to be done, honestly, in this field. We're really, really early on.
And about the case 9,
About the?
Case 9, the base screen.
Oh, yeah, the base screening is a system for, so Cas9 is fused to the deaminase, and so we can in this way mutate the amino acid, a single amino acid resolution. So we have guide RNA that are tiling the entire length of the protein of interest, and we can mutate that individual site that we want to mutate and then ask what is the contribution of that mutated site on the protein and on the cellular outcome. So, of course, if we mutate a residue in the binding pocket of this YTHDF protein, this protein are not binding M6A anymore, but it's exciting to see what else is happening there. And I know, but I have to go back to the idea that amino acids are modified, and so we are studying post-translational modification of this amino acid and see how. Probably, I don't know. I don't think we have methionine in our screen, so we cannot collaborate. But there are phosphorylation and things like that that probably are involved. Yeah.
And that's measured by your Z-score?
Yes. Yeah. I'm happy to give you more detail later on. There are different labs now at Columbia that are doing this type of base editor screen, especially in the medical campus, and they are normally used to check simply how genetic variants or even a SNP or like a mutation is impacting cancer, but they can be used as a system to interrogate a protein or interrogate whatever things you are interested in. So they are very, very powerful technology in that sense.
Okay, questions to the lunch break. Thank you so much, everyone, for the questions, and especially our speakers. Let's thank them again for the great talks.